Well, it's a wonderful night, of course, and my name is Luke O'Neill. I chair the programme committee for this event, which was a really big task, and I want to thank all the other people involved, especially Eamon Cahill, actually, who is here somewhere. But it came down to picking the keynote speaker, and we were lucky because we had a meeting in London in October, and just before the meeting they announced the Nobel Prize for Medicine or Physiology for 2011. And I went to a man from Luxembourg who did all his work in Strasbourg by the name of Jules Hoffmann. It also went to two Americans, we won't mention them because this is an ESOF event. But it was wonderful that a European had won the Nobel Prize. It was even more wonderful because he was in my exact area. I'm an immunologist. And in fact, all my work was inspired by Jules Hoffmann, it must be said. Thousands of immunologists were inspired by him. He made a discovery, a fundamental discovery in the immune system, only working on fruit flies. He's a real hero to us all. He was a fundamental biologist working on the fruit fly. And he goes on to win the Nobel Prize for medicine. Now, you might scratch your heads and wonder why that would be. Now, when we were sitting down here earlier, he asked me what language the president was speaking. He asked me, was it Hebrew? And I got thinking, what was the Irish for fly? And of course, the Irish for fly, Jules, is quillogue. So you can mention quillogue in your talk. But he discovered this pathway, this immune pathway in the fruit fly. And when the fruit fly is infected with fungus, it triggers a receptor called toll. And toll senses the fungal infection, and the fly now defends itself. Now, that was a really good paper. Next thing, we find toll is everywhere. Every species of life on Earth has this immune pathway. Now, I work on a species less interesting than fruit flies, the human. From Jules' point of view anyway, they're in humans, and these tolls drive disease. They're in MS, they're in arthritis. When the immune system goes wrong, you get nasty diseases. It turns out tolls are very much part of all that. And that discovery began with Jules Hoffmann, and we picked him because outstanding basic science. Again, you cannot overemphasize the importance of basic, fundamental, excellent research, and that's what Jules Hoffmann represents. Secondly, that he was actually a European, which of course is another big box to tick. And thirdly, he's a very, very nice man. So please join me in welcoming Jules Hoffmann, 2012 ESOF, to give the keynote address.
Thank you really very much, my friend. Yes. Thank you, Luke, for this very warm introduction. And let me start. Dear Commissioner, Mrs. Kagan-Quinn, dear Ministers, dear President Benda, dear Chief Scientist, Mr. Cunningham, honored guests. I feel, of course, very honored today to give this presentation in front of such an august audience, and I warmly thank the organizers for this invitation, in particular, of course, Professor Luke O'Neill. The title of my talk, as suggested to me by the organizers, calls for reflections inspired by my personal scientific journey through basic research in a predominantly European context. I would start by providing a background of this journey, which I will illustrate with a series of slides. I will then turn to my personal reflections inspired by this journey. I was born in Echternach, a small city on the border between two European countries, Luxembourg and Germany. Echternach was once famous for its Benedictine abbey. And this abbey was founded in the 7th century by a Northumbrian bishop, Willibrordus, who had received his profound Christian education here in Ireland. Indeed, after the fall of the Roman Empire at the end of the 5th century, much of Christianity had disappeared in the areas which were overrun by the invading Germanic tribes. Christian Ireland had been spared these turmoils and became a prime center for re-Christianization of northwestern Europe during the 6th and 7th centuries. Whereas Willibrordus influenced my young years in Luxembourg, another Irish missionary, Columbanus, was watching over me when I later moved to eastern France, which he had abundantly seeded with abbeys. Standing here tonight, I realized that I never really escaped from Irish influence. Although the earlier saints have now been replaced by secular giants, such as the local organizers of this prestigious ESOF meeting. I grew up as the son of a high school teacher in biology. My father spent his free time collecting and identifying invertebrates, and namely insects in the countryside. Early on, through him, I became familiar with this large animal group. Insects, indeed, make up some 80% of all species present today on our planet. They destroy 1/3 of human crops and put 1/3 of the human population at high risk of contracting often fatal diseases through their capacity as vectors of viruses, bacteria, and parasites. The experience of my father induced me to choose biology, and more specifically zoology, for my university studies when I moved to Strasbourg. I was fortunate to be accepted for PhD work in the laboratory of Professor Pierre Joly at the Institute of Zoology at the University of Strasbourg. Professor Joly and his associates worked on the development of grasshoppers, and these insects were at that time a major pest in Africa, which they appear unfortunately to have become again in recent years. The Joly laboratory routinely performed transplantations of hormone-producing organs or brains between individual grasshoppers and studied their influence on development and reproduction. Professor Joly reflected in one of our discussions that although he had not taken any precautions against microbes in these experiments, he had never observed any opportunistic bacterial infections following the dissections. This was the determining moment of my scientific life. We decided that I would investigate the mechanisms which explained the remarkable resistance of this important zoological group against microbes. When I started my thesis work, two types of insect defense mechanisms had been proposed in the literature. One, championed by Elia Mechnikov at the end of the 19th century, initially in Russia, later in France, was the phagocytosis, that is to say, engulfing and destruction of microbes by dedicated blood cells, the so-called phagocytes. The other, discovered after the First World War in Germany by Rudolf Glaser and in France by André Payot, was the inducible production by infected insects of antimicrobial substances. These substances could, however, not be identified at that time. During my PhD work, I established that grasshoppers rely essentially on the cellular arm of defenses, that is, on phagocytosis of blood cells. I could trace back the origin of these cells to an as yet unknown hemopoietic tissue in the vicinity of the dorsal heart vessel. When I submitted this tissue to selective X-ray treatment, as shown here, two major effects were induced. Let me just take 1 second to explain that this is a section through a grasshopper. This is the gut, this is the heart, this is the nervous system, and here we have this hemopoietic tissue. So I performed selective irradiation of the upper part and of this part here as sham irradiations. So the results were the following. One, the hormonal control of development was upset when the treatment was performed at a critical stage. Number two, the treated insects developed spontaneous septicemia without any additional challenge. Sham irradiations of other areas of the grasshoppers did not affect these parameters. Insect development was known at that time to be under the control of the molting hormone ecdysone, a steroid discovered by Peter Carlson in Germany. Carlson was a professor at the University of Marburg and had been in contact with my mentor, Professor Joly. We decided that I would join the Carlson laboratory in Marburg for some time to get familiarized with the biochemistry of steroid hormones. Over several years, we developed a fruitful collaboration in the field of biosynthesis and metabolism of ecdysone. Carlson was a distinguished biochemist who had published one of the very first textbooks in biochemistry, which became a bestseller worldwide. In addition to introducing me into the secrets of biochemistry, Peter Carlson also introduced me into the German scientific community, with which I have ever since maintained very close contacts. There existed at that time a thriving community in Europe working on insect physiology and development. One of the prominent members of this community was, and still is, Walter Gehring from Basel, with whom I became befriended and interacted frequently. Walter introduced me to the microbiologist Hans Bohmann in Stockholm, who had initiated studies on antimicrobial substances in butterflies. It was thanks to Hans Bohmann in Stockholm that we became familiarized with microbiology. I'm speaking about the European journey. Bowman and his associates had isolated from pupae of the butterfly Hylophora cecropia the very first inducible antimicrobial substance and had identified it as a peptide which they named cecropin, after the name of the butterfly. Bowman accepted to serve on the thesis defense committee of my student and wife, Danielle. I'll show her picture in the end of the presentation. And later invited her to spend some time in his laboratory to work on the isolation and characterization of antimicrobial peptides from various butterfly species. However, when Danielle came back to Strasbourg, we tried unsuccessfully to find similar inducible peptides in grasshoppers. These insects appear to rely essentially on phagocytosis for their antimicrobial defense. We then turned to large blowflies to try to isolate inducible antimicrobial substances. Eventually, we were able, from the blood taken from many thousands of challenged blowflies, to isolate one inducible antibacterial peptide. Unfortunately, we did not have the appropriate equipment at that time for amino acid sequencing of the low amounts of peptide which we had isolated, in spite of having started from 1 liter of blood of challenged blowflies. I turned to Professor John Fothergill at the University of Edinburgh, where a pioneering center in peptide analysis had been built up. With his generous support, we could finally establish the amino acid sequence of this inducible antimicrobial molecule, which we called diptericin. At the end of the 80s, we turned from blowflies to Drosophila in the hope to use the powerful genetics of this model insect to decipher the molecular basis of the antimicrobial defenses. We first noted that a microbial challenge induces the rapid appearance in the blood of adult flies of a potent antimicrobial activity. Here we prick a fly with a needle which has been dipped into a culture of microbes, and we take off the blood after various time intervals, and we test for antimicrobial activity with a very classical growth inhibition assay. Our first objective was to isolate and characterize the antimicrobial effector molecules, which were still unknown in this species. Assuming that they were peptides, following on the steps of Bowman for cecropin and those of our laboratory for diptericin, we intended then to turn to the control of expression of the corresponding genes. Question 2. Thirdly, we wanted to address the question as to how the fly recognizes infecting microbes and whether it can discriminate or not between various types of microbes. Over several years, we could show that Drosophila produces at least half a dozen families of small-sized antibacterial peptides, which are shown in this slide. So the center of production of these molecules is the fat body of insects, which is equivalent to the mammalian liver. And here we have diptericin, which is active against gram-negative bacteria, drosocin also against gram-negative bacteria. Cecropin and diptericin are molecules which we cloned through homology after the data which were produced in the butterfly by Hans Bowman, and defensin, which is anti-gram positive. One essential point here is that those molecules are secreted into the blood where they reach extremely high concentrations. And this secretion this model has been one of the motors for our attempt to look at their therapeutic interest. A hallmark in these studies was the isolation and identification of the first antifungal insect peptide with similarity to an antifungal peptide from plants. For this, we teamed up with Willem Brokker, the professor at Leuven University in Belgium, a specialist in plant antimicrobial defenses. We call the newly isolated Drosophila antifungal peptide drosomycin. It's shown here. It's an extremely interesting molecule. It has 4 disulfide bridges, and this was actually the lead molecule for our company, to which I'll come back in a second. It is of interest to mention here that 3 years ago, a group in Nijmegen, in Holland, found that the human skin produces a drosomycin-like peptide, pointing to the conservation of some aspects of antimicrobial defenses between flies and humans. And I will, of course, come back to this point later. Significantly, when we cloned the dipterosin gene, we noted that its control regions resembled those of mammalian genes which are upregulated in stress or defense reactions by a central regulator protein named NF-kappa B, a nuclear factor isolated a few years earlier by David Baltimore in B cells, hence the name. So let me just explain this in a few words. So NF-kappa B, we have 5 members of this family in our own defense system. This is the molecule. It is present in the cell, but it is not active because it is blocked by an inhibitor. And this will become apparent. This is David Baltimore who found the molecule 96. And this is NF-kappa B here. Those are studies which were produced far later than after the discovery of NF-kappa B. And they are blocked in the cell by binding to an inhibitor, which is given here by the name I-kappa B. Now, the beauty of the system is that to immediately induce a response to microbes, you don't have to synthesize anything new here. You just try to take off the inhibitor I-kappa B, and then NF-kappa B goes into the nucleus, and there it around, so please, it went, yes. And here, as a dimer, that's to say 2 of these molecules sit around DNA in the regions which have those sites, which we found in the dipterosin promoter, and then they control the expression of the cognate genes. Now, it turned out that such a factor, an NF-kappa B-like molecule, had been identified in Drosophila in Germany in the 80s by Christian Nusslein-Volhard and associates during a genetic analysis of early embryonic development. This is Christiane Nusslein-Volhard. She made wonderful work on the development of axis, dorsoventral, namely axis in Drosophila. She was awarded the Nobel Prize in 1995, together with Erich Wieschaus and with Ed Lewis. Indeed, the studies of Nusslein-Volhard and her colleagues had identified a gene cascade which controls the dorsoventral axis of the Drosophila embryo. The cascade is triggered by transmembrane receptor protein, which Nusslein-Volhard had named TOL in reference to a bizarre appearance of a mutant embryo. In fact, those studies were done by feeding the parents on a mutagene and looking at the development of embryos. They scored, their colleagues scored for embryos which would not develop, and those embryos then would have a certain appearance, certain phenotype, and some of these then would be called, as in the slide here, would be called TOL because of the appearance and so on. So basically, and as I don't have much time, basically what they found, there was NF-kappa B bound by I-kappa B, inhibited by I-kappa B from controlling gene expression. When I-kappa B was released from NF-kappa B via phosphorylation, I'm going to go into detail, it went into the nucleus to control expression of developmental genes. Now, TOL itself became activated in the system after a complex cascade of proteolytic enzymes cleaved a cytokine or growth hormone here, which goes by the name of Spätzle. Again, those names were given in reference to the phenotype of the embryo. And I mean, for many of us in the audience, Spätzle, the phenotype is apparent. And they also have a noodle in their system, which is not shown in this slide. Now, these genes were maternally expressed genes. And the question arose, could this cascade be used for the induction of the antimicrobial peptides here? So first of all, we showed that these genes were expressed later in development, and secondly, that the expression was strongly upregulated by an immune challenge. So over the next years, our group set out with a newly hired Drosophila geneticist, Bruno Lemaitre, to check whether parts of this cascade of genes were also involved in the immune defenses. This led to the demonstration in 1996 that the TOL pathway controls indeed the expression of the antifungal peptide drosomycin as shown here. So this is the TOL pathway. Fungi and gram-positive bacteria will activate this system, not directly, I'll come back to that in a second, and then TOL will induce an intracytoplasmic signaling cascade, which will allow NF-kappa B to go into the nucleus and control the expression of the antifungal peptide drosomycin and many other genes. This pathway significantly contributes to the resistance to fungal infections. This is a fly in which, this is a toll mutant fly. It's deficient for the toll gene, and it has been infected by Aspergillus, and you see that this fly is overgrown by the fungus, by the hyphae of the fungus, showing that the interfering with toll results, at least in the fly, results in a terrible disease which kills the fly. This is a paper, this was a cover of the Cell paper which was published by Bruno Lemaitre, two students, doctoral students, Jean-Marc Reichart, who is now the director of our laboratory, and myself. Now, in contrast, we found that antibacterial defenses, and namely the expression of dipterysin, were mostly independent of the toll pathway, but were controlled by a second set of genes, which we refer to as the IMD pathway. And here we have a direct interaction of gram-negative bacteria with this receptor, and then activation of a member of the NF-kappa B family, production of antibacterial peptides, dipterysin cecropin. So I'm summarizing here the effort of 6-7 years of work. Initially, we found that dipterysin was not dependent on the toll pathway, which was a very black moment in our laboratory because we didn't have drosomycin in our hands at that stage, but this is explained in detail in the publications. Now, in 1991, I had contacted and visited Professor Charles Janeway at the Department of Immunobiology of Yale University, and this is Charlie Janeway. He died, unfortunately, 8 years ago. And Al Nazikowicz at Harvard Medical School, who were both interested in the basic mechanisms of immunity, and namely in the recognition of pathogens. In particular, Charlie Janeway focused on the requirement of the innate immune system to boost the adaptive immunity. Now, this needs some explanation. Let me give this quickly here. We humans, have two responses to immune defenses. One is referred to universally now as innate immunity. This is a phagocyte. It's an immediate reaction. It has no memory, and it is common to all organisms. A second response is the adaptive immunity. It is slow. It takes several days to get in place. It is found only in vertebrates. It has appeared with lymphocytes, which are specific for vertebrates, and lymphocytes have appeared probably in ancient cetaceans about 450 million years ago. Those lymphocytes can produce a large repertoire of receptors in the millions. They also can produce memory cells. Lymphocytes can become memory cells, and consequently, this allows for vaccination. Now, at that time, let's say from the early 90s on, the idea was that the adaptive immune system, symbolized here by a naive lymphocyte, which has to be activated, that this requires an input from what was called antigen-presenting cells, that is to say, cells of the innate immune system. I'm showing here Ralph Steinman, who was co-laureate of the Nobel Prize and has worked for 30 years on these cells. Now, the activation, it was supposed, occurs when microbial fragments interact with these cells, either dendritic cells, macrophages, or so on, and then activate NF-kappa B. But the receptor here was not known. The result was supposed to be, on both sides shown here, an immune response of an innate immune response, that's to say induction of antimicrobial peptides on one side, as we had it in Drosophila, and in mammals, in addition to this, activation of the adaptive immunity by the innate immunity. So this was really a crucial question in the mid-90s, because as you see, our defense depends on the innate immune system. Now we know and we have understood that largely this on the adaptive immune system, lymphocyte, that it takes a boost from the innate immune response. Now the innate immune response is dependent on this hypothetical receptor, several receptors. So all of our defenses against microbes somehow, at least to a large extent, depend on these receptors on innate immune cells. Now, the receptors were not known, as I mentioned. So Janeway, let me say this again, he postulated that a dedicated transmembrane receptor protein was present on the cells of the innate immune system. Now, we set out to collaborate on this, and to make a long story short, we engaged with Charlie Janeway and Elna Zekowitz in a collaboration soon to be funded by a major grant from the Human Frontiers in Science program, which has its general office in Strasbourg. And 1 year after our demonstration that the transmembrane Drosophila toll receptor is required for antifungal defenses in flies, that was in '96, the Janeway laboratory identified a human toll homologue. They showed in transfection experiments that the human toll could activate NF-kappa B to express some of the genes required for boosting adaptive immunity. Still 1 year later, in 1998, Bruce Beutler, then at the Scripps Institute in La Jolla, now in Dallas, identified in mice the gene encoding the receptor for the classical bacterial cell wall component, which is called lipopolysaccharide, LPS. It turned out that this gene encodes a transmembrane protein similar to Drosophila toll and to the human toll homologue characterized by Janeway. As a short epilogue, we now know that humans have a dozen distinct functional toll-like receptors which respond to various microbial cell wall signatures or to viral nucleic acids. They convey a potent and mandatory first-line defense, the innate immune defense, and in parallel activate the adaptive immune response, which is once more shown here. So these are the two types of toll receptors. One group which is on the cytoplasmic membrane, on the cell membrane. They bind essentially to lipopeptides, to lipopolysaccharides, also to flagellin. And then another group which recognizes nucleotide sequences from viruses or bacteria, which is in the cytoplasm in the endosome compartment. In particular, some lymphocytes are stimulated to produce antibodies, as shown here, to produce antibodies, or to become cytotoxic and to destroy infected cells, thus restricting the spread of the infection. And as mentioned repeatedly, the system also provides the production, controls the production of antimicrobial peptides, which is our first line of defense. And there are a large number of adaptor proteins, a large number of 5 adaptor proteins here, which have been worked out in detail by Luke O'Neill here in Dublin. In Drosophila, subsequent work performed in Strasbourg, Stockholm, Paris, and also in the United States, in Korea and Japan, has unraveled major players of the signaling cascades, which lead from the recognition of microbial cell wall components via the membrane receptors to the activation of NF-kappa B, that is to say, to the phosphorylation of the inhibitor, which is then going to be to release NF-kappa B, will become degraded and then go into the nucleus to control expression of genes. Now, these signaling cascades are very complex. And here in Dublin, a lot of work has focused on this. A lot of work. I'm sorry, something happened. Is that it? Luke, can you protect me? That's the immune system. That's the immune system. That's still from the show. That's still from the show which we had earlier. So, well, that's because I did not agree with the compliments I was going to pay to you for the signaling cascades, which have been worked out so elegantly here, and which were also worked out to a large extent, I wouldn't say elegantly now, in the fly. And let me just say that a striking outcome of these comparative studies was the realization that the basic mechanisms underlying innate immunity from the receptors to the control of gene expression share significant similarities between flies and humans, not only in terms of the identities of the various players within the cascades, but also their respective places within the circuitry as shown in this slide. You wanted to see the slide. Yeah. So what I've shown here, I'll be quick on this. It's the take-home message. So this is a cascade, a few of the members of a cascade of the toll pathway in the fly, and this of the IMD pathway in the fly. We have the receptor, we have adapter proteins, we have kinases, and we have, of course, the system of NF-kappa B, with which I believe we are all familiar now. Now, I've put here the TLR4 system, which is a human or mouse system responding to the lipopolysaccharide, as indicated here, and the TNF receptor pathway indicated here. Again, we have receptors, we have kinases, and we have activation of NF-kappa B by phosphorylation, by removal of this inhibitor. Now, I've chosen the same colors here so that you can see how the situation is similar. But this is true not only for the ones which are presented in this slide. In each of these cascades, there are between 12 and 20 members at least, but it is valid for all those members. So this was really an enormous surprise to us. I mean, we would never have believed in the 60s, 70s when we started this work, nor would anyone have believed in the mammalian system. This had not been worked out yet at that time. No one in the medical faculty would have believed that the systems were so close in evolution. So taken together, the results obtained in Drosophila, mice and humans, have put innate immunity, a long neglected and underestimated facet of our antimicrobial defenses, into a new perspective. They've also underscored the role of toll receptors, a new class of receptors for microbial signature, cell wall components, nucleic acids for microbes, but also, and importantly, abnormal self released, for instance, by dying cells in our system. Significant new developments for medicine are expected to stem from this data. This is an evolutionary slide in which I've put the invertebrates here, which all rely on and only on innate immunity, and the vertebrates, which rely on innate and adaptive immunity, thanks to the development of lymphocytes in fish around 450 million years ago. Now, what we can now conclude is, as we have the genome of the sea urchins available, and of worms and so many other groups, we now can say that from toll to NF-kappa B, the system is conserved. The system has evolved probably around 1 billion years with the appearance of multicellularity. So the last point here I wanted to make is that now it has appeared over the last years, over the last 5 years, it has appeared that toll-like receptors are involved in many aspects of medical interest. For instance, they play a role in recognition of several key adjuvants. That is to say, they play a role in vaccination. And we are all aware that vaccination is one of the major problems nowadays worldwide. They are also involved in autoimmunity. Autoimmunity is, again, it's a problem. It's an increasing problem in developed societies. And it essentially, let me say, in a certain number of instances, it is dying cells, chromatin, DNA, which is recognized by toll-like receptors and is at the origin of autoimmunity. This is also true for allergy. It's true, of course, for inflammation, which is one of the major conditions in the developed world. Now, inflammation also is mediated via TLR, so toll-like receptors. Infectious diseases, of course, and more in immunotherapy. Ladies and gentlemen, after having taken you through this journey of nearly 50 years, I would now like to present some personal reflections. A first point which I would like to make is that we started our research simply driven by curiosity. Here was an important zoological group, the insects, which appeared to be highly resistant to infections. Why? We had no precise goal beyond the desire to understand the mechanisms explaining why this was so. We were sponsored and supported by the University of Strasbourg and by CNRS, the French National Research Agency. Moderately, I may say now. Significantly for that period, we were not asked to indicate which milestones we wanted to provide, within which time frame, what applications we were hoping to generate, what networking we were planning to develop, which industrial partners we had contacted. I sometimes feel that this was a blissful time. There was a great confidence in science and a global belief that whatever the field and the questions, any new scientific knowledge would eventually have positive outcomes for society. The recently deceased great French Hellenist and professor at the Sorbonne and at the Collège de France in Paris, Jacqueline de Romilly, once wrote, "Comprendre est ma joie, comprendre le plus possible, de tout, toujours." I translate, "Understanding is my delight, understanding as much as possible of everything continuously." I'm convinced that the desire of understanding is the primum movens for all of us in science, our driving force in our individual journeys. I would like to argue that our society should continue to support, to a significant extent, research which is purely based on curiosity, even in the absence of perspectives of applications when the work is started. It is, of course, not my intention to oppose basic research to applied research. We obviously need both in our society. Let me shortly mention that here that we had the opportunity ourselves, as I already said, at a given stage to look into the therapeutic potential of some of the novel antimicrobial peptides from insects, and namely of the antifungal peptide drosomycin. We started a company, Entomed, Entomology Medicine, in Strasbourg, and over a period of several years, this company had employed 40 persons. And I'm happy for this contribution which our research made, at least temporarily, to our society. The efforts are for the time being stalled because of the high price of producing these molecules in high amounts. But this might change in the future as new methods are being developed. A striking aspect in my journey, and this is my second reflection here, relates to the fact that the concepts and methodologies on which we relied in our research have constantly evolved. In other words, we have been dependent all along this journey on the progress of other disciplines. The advent of molecular biology, namely, has profoundly changed our research. Similarly, to give a second example, the progress of mass spectrometry analysis, coupled with the availability of the Drosophila genome in 2000, allows now the identification of an antimicrobial peptide from a pool of 10 flies, as opposed to the tens of thousands of flies in the early 90s. The university lectures which I attended during my student time helped me in asking good biological questions, but they had little direct relevance for our work 20 years later. The progress in science has been spectacular in nearly every field over the period of my journey, and there's every reason to believe that progress will continue at a similar, if not a still more rapid pace during the next decades. This is, of course, a challenge for everyone in science. Keeping pace with the evolution of science in one's own field and in neighboring fields, which is a requirement for interdisciplinary research, will be more and more challenging, particularly for smaller groups. In addition, such groups can often not afford the sophisticated equipment which more and more comes to dominate our research work. What can our institutions do to help young scientists to have access to continuous training and updating during their research? And further, to give them continuous access to rapidly evolving, sophisticated and expensive equipment. A positive answer to these constraints has been the concept of large institutes of research, which, in addition to performing first-class investigations on their own, provide service and teaching to smaller and less integrated scientific groups. A prime example of this is the European Molecular Biology Laboratory, from whose expertise and equipment our group has considerably benefited over many years. Another highly positive endeavor are the courses which are increasingly provided at a European level in many fields. Again, referring to my personal experience, EMBO is being highly efficient in this respect. At national levels, significant efforts are being oriented in the same directions. A great example in this context of my personal journey is the Institute of Genetics and Molecular and Cellular Biology created in Strasbourg by Pierre Chambon to the benefit of the whole community. My third reflection relates to interdisciplinary research and collaborations between laboratories which are not necessarily working on the same topic, but which ideally have complementary expertise. Although such directions can be taken fairly easily through the initiative of respective groups, as was the case For us, European institutions have been very helpful in fostering networking over the European Union. I remember with pleasure and gratitude one such network on malaria research at the initiative of Fotis Kafatos at the European Molecular Biology Laboratory in Heidelberg. This network brought our group working on Drosophila into enriching contact with mosquito groups from Southern Europe, Germany, and the UK, with whom we would probably not have interacted spontaneously without the network of Fotis. The European framework programs have undoubtedly had a beneficial impact, particularly by providing fresh funds and equipment in a context of well-defined and well-structured inter-European collaborations. I understand that Horizon 2020 will attempt to create even better framework conditions, as we just heard, for research and innovation and fund the best frontier research as well as close-to-market applied science, as recently stated in editorial in the journal Science by Mrs. Geoghegan-Quinn and repeated a few moments ago here. I would further argue that one of the most efficient moves to foster high-quality research in Europe was the creation of the European Research Council. Selecting by transparent procedures excellent individuals without imposing a research theme and further providing significant financial support is without doubt one of the best investments for the future of science on our continent. I would love to see the ERC operation increase in volume and be adopted in its principle on national levels. One last reflection. When I started doing research in the 60s, there was a general enthusiasm and confidence in science within our society. There was also real respect for universities and research institutions, should even say for professors. Society has, of course, evolved during the decades since, and science has immensely progressed, and de facto, by progressing and accumulating immense amounts of new knowledge, has become so complex that most of our fellow citizens feel overwhelmed or lost. Science still enjoys a relatively positive image in the general public, as repeatedly indicated in polls. However, a significantly large and vocal group of citizens have developed a marked level of distrust towards scientific research. I find this to be particularly true in Europe and specifically in France, in areas such as genetically modified crops, vaccination, research on stem cells, electromagnetic waves. There's a long list. Regaining the trust of these opponents will not be an easy endeavor. Clearly, the general media can play a role here, and we have to make every effort to foster good contacts with serious media and get the public interested in our research, while avoiding, of course, to oversell our results, which could only feed the distrust. But obviously, the future of a confident relationship between an ever-expanding scientific world and our fellow citizens lies with the education in science of the young generations. This education should not only be centered on the mandatory acquisition of scientific knowledge, it should also make young people familiar with the ways in which scientific knowledge is generated, checked, counter-checked, with the limits of given experimental approaches, with a sense of using probabilities, of using cohorts, with the interactions within the scientific community as a whole. Above all, it is our unique responsibility, beyond the process of training, to get young people interested in science, and even much more, to make them feel enthusiastic about science. My last words will consequently go to the younger persons in the audience who have just started a scientific career or are pondering whether or not to take that direction. I wish to remind them that it is nearly beyond imagination how in most scientific areas, particularly in life sciences, the world has changed within the last 50 years. Concepts and methods are now available nearly in any laboratory of which my generation could not even dream when we started our scientific journeys. Funding will, of course, never match our expectations, but it is still incredibly higher today than it was a few decades ago. And last but not least, the perspectives of finding a job with a scientific background, namely with a good PhD, have so markedly increased and diversified. to give a last example from my journey, of the numerous doctors formed in our laboratory, only 40% remained in academia, the others having taken up jobs in companies, administrations, public relations, in media, scientific journal, or other areas, and they all have done well. Likewise, whereas it was a rule in our early years that most PhDs would stay in their country, this was particularly true for France, 1/3 of those who got a PhD in our laboratory now work in other European countries or in the US. I sincerely believe that as scientists or future scientists, they should strike an optimistic note. They will work at the forefront of knowledge, which is one of the most privileged places in society. And they will do their investigations at the time in human history of great discoveries with enormous potential. Indeed, I envy the young people who are starting their scientific journeys today, and I wholeheartedly wish them great success. Thank you very much for it.
Thank you very much, Jules. I might wrap up the event now and say it feels like the start of our summer holidays, right? There's all this fantastic science coming. So make sure you engage with it and make sure you take part. Go to the pub. As Dara said, the Guinness trade needs it. But let me ask you once more to give a big round of applause to the Higgs boson of immunology, Jules Hoffmann.